Hello, and welcome to the Badger Talks podcast, the podcast that shares interviews with experts from the University of Wisconsin-Madison community about their work, research, and a little bit about what they're like as people, too. I'm your host, Buzz Kemper. Today, we're focusing on bacteria that evade immune systems, and I'm happy to have as my guest Dr. Suzana Salcedo, Associate Professor at UW-Madison's School of Veterinary Medicine. Suzana, thank you so much for doing this program. This is a topic about which I know nothing, so we're going to have to kind of start from the basics and move up. But the first thing I want people to know is that you're a fairly recent transplant to the United States and to Madison. It's been about a year and a half, correct?
That's correct. Thank you very much for having me. It's a pleasure to be here. So yes, I arrived summer 2023 when I arrived from France, where I was directing my research group there for about 10 years. Yeah. And so, I'm a new arrival in Madison.
Yeah, you've studied in Portugal, in London in the UK, and then you've been doing research in both Marseille and Lyon, France.
That's right. First, I was hired as a junior researcher in the south of France.
Okay.
And then I managed to get a big funding grant to support starting my own group. And then I moved to Lyon, where I directed my research group for 10 years.
Okay. And you're in the School of Veterinary Medicine, but a lot of what you're doing is also research that affects human beings as well. So give us the basics of how these bacteria work and what it is that you're studying.
Okay. So we do work with a number of bacteria that cause disease both in animals and humans. And actually all of these diseases are connected, right? So when we study human disease, we also have to take into account animals and the environment. Mm-hmm. And so we are trying to understand how these bacteria can infect host cells, animal cells or human cells, either by invading them and going inside and hiding from the immune system and multiplying and sometimes causing cells to die, and then leading to disease. So this is what we are trying to understand.
Okay. And when you say hiding, that means what? They're somehow getting away from any kind of immune defense system that might be able to destroy them?
Exactly. So the bacteria that I work with are really good at hiding from the immune system, at least during a period of time. And so when they get inside the cell, they're inside this little cocoon that protects them and keeps them hidden from detection from the immune cells. And we're trying to understand how they do that. And at the same time, they are getting time to multiply and to grow in big numbers inside these cells so that they can get out and infect other cells. And then this is how the infection spreads, right?
Wow. And is this an evolutionary thing? Like these bacteria, they at one time were in a form where they were being attacked by the immune system, but they've evolved so that they now can evade the system? Is that correct?
That's an excellent question. So it is true that some of these bacteria do have what we call virulence factors, so genes that allow them to become more dangerous to humans and animals that they acquire during evolution. And we are particularly interested in a set of genes that encode for a system that is called a secretion system. It is like a nano syringe with a needle, so really tiny syringe and needle that allow bacteria to inject proteins inside the host cells. And these proteins is what my lab is studying. These proteins can be toxins, for example, and they are causing disease or allowing the bacteria to hide from the immune system.
Evolution, these kinds of changes are a double-edged sword as the bacteria can evolve and make itself more defended or better way to hide from the immune system. You would think that the immune system would also be upping its game. Is that correct, or is it kind of a one-sided war at this point?
Well, we're not at the same speed, I guess, right? And that's what we try to do when we introduce vaccines and we try to teach the immune system how to fight these bacteria. But some of these bacteria, for example, one of the ones I'm studying, in some animals, it doesn't cause disease. So it seems to have learned to live with this host for a very long time. And when it finds itself in a human, which is not its natural host, then it causes very severe infection. And so the humans are not prepared to handle this type of bacteria, but some of the animals in the environment are and can coexist with this bacteria for prolonged periods of time.
Okay. And so, this is so fascinating. So some of these bacteria, they will start in animals. I know that humans are animals too. So let's say non-human animals, and then they spread to humans. And I imagine that they manifest themselves in different ways in a human than they would in like a dog or a cat or whatever. And so does that make it more complicated for you?
It does, and it's very important that we're using the right models to study different steps of the disease, right?
Okay.
And so, for example, just to give you one precise example, one of the bacteria we study is called Brucella. It causes brucellosis. So this is pretty much been eradicated quite successfully in the United States.
Okay.
It usually infects livestock like cattle, sheep, and occasionally dogs as well. But it's still present in wildlife, for example, in the greater Yellowstone National Park. And sometimes there are spillovers still to herds of cattle in the region.
Okay.
Right now, there's quite a few herds in the quarantine.
So when you say Yellowstone, are we talking about bison or elk or
yeah,
what?
Exactly. So
Okay.
the bison, and more recently, it's more the elk that are a problem, that are infected with Brucella and have brucellosis. And as you can imagine, it's really hard to contain these animals and control this disease.
Right. Yeah, they're wild animals.
Yeah.
So what is Brucella? Is it Brucella? Am I saying it correct?
yeah.
Brucella. What is the disease?
So this bacteria in cattle, for example, it can lead to abortion in pregnant female animals, milk reduction, and the bacteria is shed in milk. So then there's a danger for humans, because if we consume unpasteurized milk, raw milk that is infected with Brucella, then we will get infected. And it causes a very severe infection in humans that has to be treated with two antibiotics for a prolonged period of time. And so we don't want that. And so the United States has been very successful of eradicating Brucella and brucellosis in humans and in cattle. But sometimes you get this spillover from wildlife that is very hard to control.
Okay. And in humans, what are you said you told us some of the symptoms in animals, the aborted childbirth, the milk being affected. What are the symptoms in humans? Are they the same?
So in humans, Brucella can pretty much infect any organ of your body. So if it doesn't get, at the beginning of the infection, you get a very non-specific set of symptoms. Very bad cold with what we call undulant fever. So fever that comes and goes, and you feel really sick and weak, headaches. So it's very non-specific. So it makes it quite difficult to diagnose if you're not in an area that is used to seeing brucellosis in humans, right? And then if it does not get treated, you can get severe complications like chronic infections, arthritis, or even more severe like neurobrucellosis or infection of the heart and cause severe endocarditis. The good news is that treatment is efficient. It's long, it requires antibiotics, but it does work. So it has to be well diagnosed in humans.
Okay. And does your research help these treatments become developed and refined?
So we are more focused right now on trying to improve strategies to vaccinate animals.
Okay.
So that we can prevent passing it to humans.
Okay.
Because if we can control, and that's that concept of the One Health, if we can control the disease in animals, we don't have to worry as much for the human health, right?
That has to be daunting, though. How do you do this? How do you get a vaccine to a wild animal that's running around in a huge, many thousands of acres park?
This is an excellent question. So already if we can protect cattle a bit more efficiently, then we can maybe allow the wildlife not to be treated for the moment. But there are a lot of groups all over the world trying to get vaccines that could be either shot from a distance or maybe even put in a spray or in the water. So there's a lot of work that has to be done to improve that. So we're not even close to doing it, but it would be really exciting if we could control the disease in wildlife.
Oh, wow. Is your lab involved in that kind of research as well, like how to deliver these vaccines?
No,
or is
we're still...
That's
further down the line.
That's down the road, okay.
We're still working more with cell models of the different types of animals that get infected and trying to understand what could we block from this bacteria that would prevent the infection. So not only thinking of vaccines, but potentially treatments, because maybe it would be easier to treat And in that case, we could maybe put a treatment in the water or in the food, for example.
Okay.
So that's what my lab is doing, is studying at the cellular level what is going on. So that nanosyringe that is very important to cause disease, if we could block it, then Brucella would not be a problem anymore.
Okay.
So just to give you an example.
Yeah. And is Brucella, would you consider that to be the most sort of dangerous, the most, the thing that should be focused on the most in terms of these kinds of bacterial diseases that can affect both animals and humans? Or are there others that, you're shaking your head.
There are many.
Many, okay.
So Brucella is very interesting because it has a very high pathogenic potential. So it actually used to be considered a bioweapon in the United States.
Oh my gosh.
Only a few months ago, it got removed from the list because 10 to 100 bacteria sprayed in a room would be enough to cause disease in any human. So it has a very high pathogenic potential, and it also is very good at hiding from the immune system and inside the cells. So it's a good model for us to study. And we can learn a lot about this type of bacteria, but also about our immune system and how it responds to these bacteria. So maybe we can also improve the way our immune system is fighting them by studying it. So it's really a good model as well.
Wow. Yeah, that's has huge potential for both animal and human health. And I guess that's why you do it. What got you into this in the first place? This is this something that young Suzana thought, hey, here's a thing I want to do when I grow up?
Yes, it is actually.
Good. Okay, great.
Well, it started when my dad at some point gave me a book about cells and microbes and microscopes, and I just discovered this invisible world and become fascinated with microbes. And then as I went to university studying microbiology, I really became passionate about bacteria and how bacteria interact with each other, because they also have to fight each other. For example, our good bacteria that are in our body, right? Yeah. That protect us, the bacteria that cause disease have to fight them and win this battle in order to spread in the host. And so I became fascinated about bacteria and the immune system and how cells work, and just continued on and never stopped. And so I kept just increasing the danger level of the bacteria I was working with.
Okay, keep upping the danger level. Wow. Well, I mean, yeah, good for you. Go for the high stakes stuff. Where did you grow up?
I grew up in Portugal.
In Portugal.
Yeah, okay. I was in the U.S. until I was about 5 years old.
Okay.
So I was born in the U.S.
Oh.
And then my parents moved to Portugal, and this is where I grew up and where I studied microbiology.
What is the most exciting thing, if this is even a fair question, but what is the thing that you've come across in your research, either recently or farther back in history, that you found the most fascinating or exciting or maybe surprising?
Okay, there are two things that we're really excited about right now. One of them is on Brucella. Okay. And because we have discovered two new proteins that get injected into the host cells during infection by these nanosyringes. And we think that these proteins may be involved in causing abortion in pregnant animals and sterility in the male animals. And so we're really excited about that, because if there's a specific group of proteins that are doing this, then maybe we can have a chance to stop this from happening and prevent abortion in pregnant animals and also in humans. Okay. And so we're very excited about this study.
Yeah. And what is that bacteria? What is
this? Is Brucella.
That is Brucella.
This is in Brucella.
Okay.
And the other project that we have that we're very excited about has been really to apply all this knowledge we got from Brucella, which really likes to live inside the cells, to another bacteria that we thought was outside cells most of the time, causing disease. This is called Acinetobacter baumannii. And so this is one of those bacteria that are a problem in hospitals that are very resistant to antibiotics. So we're running out of ways to treat them. And so patients that get hospitalized with surgery, or even during COVID when patients were intubated, this is one of the bacterial infections that was a problem. Okay. And so we started studying clinical strains that we got from hospitals, and we realized that some of these Acinetobacter are capable of invading and going inside the cells. very much like what we were seeing already with other pathogens. And so we are, and this was happening in some strains that were very dangerous in clinics. So now we are trying to figure out, and using all our expertise from studying bacteria that like to live inside the cells, we are trying to understand how Acinetobacter are doing this, what genes they are using, and what happens to the host cell. Can they see them? Is the immune system detecting them? And what can we do to stop this from happening?
Okay.
So we are very excited about that.
Are Acinetobacter and Brucella new things, or have these been with us for a really long time, but we're just now discovering how to deal with it, or they're evolving into different strains? How do we view the timeline on this stuff?
Brucella and brucellosis, we know, is an ancient disease. Okay. Even from bones that we've recovered, we can see we can detect DNA from Brucella infections. So it's been around for forever. And humans have always been confronted with brucellosis throughout history. So it's a disease that has been almost impossible to eradicate, probably because it's so widespread in wildlife. Right? It can infect marine, terrestrial mammals, and now we've actually discovered them in frogs as well, so amphibians. So it is really present worldwide. Acinetobacter baumannii is what we call an opportunistic pathogen. It is not a big problem, and it wasn't a big problem. It's probably been around in the environment. We don't really know its natural host. But as it became more resistant to antibiotics and started getting those genes that prevent antibiotics from working, then it became a big problem in hospitals. And so it's mostly impacting patients that are already very sick in hospitals, either intubated, severe burns, or prolonged hospitalizations. And so this is really what Acinetobacter baumannii is doing. It's a more recent problem associated with the developed world and widespread use of antibiotics.
Okay. Wow. Thank you so much for doing all this. I mean, not only doing this interview, but doing all this research. I mean, this is really having an impact on animal and human health. It's so important. Before I let you go, just two more questions. One is there anything that you would like to say about your research, but perhaps I haven't asked the right question to get to it?
Oh, you asked great questions. And actually, talking to you, I had an idea of a study that we need to do, taking more into account the evolution and maybe looking at some older strains.
Oh, wow.
So thank you for that.
Glad I could help.
It's the mind of a scientist is always thinking and getting new hypotheses.
Wow.
No, you've done a great job. You've asked great questions.
Okay, thank you. And then the last question is, when you're not involved in all of this heady research and all this, incredibly difficult and arcane work, what do you do to unwind? Do you have a hobby, or do you do anything for fun, or is this your life?
No, I do. I have two kids, so this cannot be my life.
Okay.
Right? So I love spending time with them and my family. And really, my favorite way to escape is to just go hike as far as way possible, ideally without a phone, without computer, without internet access. And this is really when I find myself surrounded by nature and just the sound of birds and water running. That's when I can really relax and kind of switch off.
Yeah.
That's one of my favorite activities.
Terrific. I'm right with you on that. I love being out in nature and just hiking around, and sometimes I do bring my phone because I'll be on a trail I don't know, and then I have to use my All Trails map to know when I get to a 3-way split. I have to know which way should I go so I don't end up getting lost. And that is a great way to clear one's head. Suzana Salcedo, thank you so much for this conversation. I appreciate you coming in.
It was a lot of fun. Thank you.
You've been listening to the Badger Talks podcast. I hope you enjoyed my conversation with Dr. Suzana Salcedo, Associate Professor at UW-Madison's School of Veterinary Medicine. Please browse our previous episodes for other topics that may be of interest to you. Badger Talks podcast is a creation of UW Connects and produced at Audio for the Arts Recording Studios in Madison, Wisconsin. Our music is composed by Bill Purdy and performed by the UW Marching Band. I'm Buzz Kemper. Thank you for listening.